Hello, I'm Jill O'Donnell-Tormey, CEO and Director of Scientific Affairs at the Cancer Research Institute, and I'd like to welcome you to the press briefing for the 4th International Cancer Immunotherapy Conference. And this obviously was a press briefing that has been scheduled, and we're very happy to have with us Dr. Crystal Mackel from Stanford, Dr. Nina Bardwaj from Mount Sinai, and of course the big news of the day beyond the conference is that we have Dr. Jim Allison here from MD Anderson, who has just been awarded the Nobel Prize. So this is a big day. Well, before I let Jim make some comments, I'd just like to preface this, that Jim is the head of the CRI Scientific Advisory Council, and he's also a fellow of the American Association for Cancer Research and a former board member. I think this meeting, as I said in my opening, has always been about science. It is not late-breaking phase 3 clinical trials that are being reported here, but it's about science, the biology, and about the mechanisms of immunotherapy. And I think no better can we epitomize that, and what we're hearing today is we talk about what Jim Allison has done. Jim, as I have said before, is a dyed-in-the-wool immunologist who never set out to hope to cure cancer, but rather just was trying to understand how T cells worked. And it is really that being an outstanding scientist with an inquisitive mind and a persistence, because you have to remember when Jim discovered CTLA-4, nobody thought immunotherapy, except maybe at the Cancer Research Institute, but besides that, no, nobody really thought that immunotherapy was going to become the 4th pillar of how you treat cancer. And certainly pharmaceutical companies were not interested in touching immunotherapy. And it is Jim's persistence that, and he'll tell the story, I'm sure, that really got at least some pharmaceutical company to be interested in and to take it on and to show that you can take a basic discovery into clinic that can benefit patients. And I think Jim is just, you're the poster boy of that and about great science. So Jim, congratulations, and please make some comments.
Okay, thank you. Thank you, Jill. And I think that it's really appropriate that this comes during this meeting with so many of my friends and colleagues from, the last 20, 30 years working on these issues here. And in fact, group of them came to our room at 6 in the morning and brought a couple of bottles of champagne. So we're having a good time now. But seriously, I mean, Jill's right. The Cancer Research Institute is the longest, I guess, institute around that's been supporting the idea that the immune system could do something about cancer. And of course, when I was in graduate school, I became aware of the work of Lloyd Old, who was, of course, a champion of the CRI and led it for many years. And I had the chance to meet him after I moved to Sloan Kettering, and as did Pam Sharma. We both learned a lot from him that as I'm sort of making the transition and to do more therapeutic work, some of the statements that he made, like learn something from every patient, that's really important now, and it's something. Maybe I'll go back to the start in a minute, but one of the things that I think that we're facing now is there's somewhere in the order of 2,000 clinical trials going on now with checkpoint agents in combination with something else. And that something else is usually chosen just because a company owns it or because for some reason, somebody decides it's a good idea, but there's almost, in very few cases, has there ever been a combination that's based on data. It's based on understanding, what happens when the patient, when the drug, confronts the cancer and the immune system. And there's just not enough patients to mess around like that. And what I think that we ought to be doing, what Lloyd said was every patient, not necessarily every single patient, but if you start a trial and you're going to, do a combination, and whether you get a clinical signal or not, you really need to get samples from that patient and study them and see, if there's a good signature, because we know a lot now about what a signature of a good response looks like. But, you can understand something about a combination by knowing what didn't happen, and then rather than just discarding it and going on to agent C or whatever, you make some well-educated guesses. Anyway, that's my political statement. But having said all that, I mean, Jill's right about the fact that I was really just trying to understand the immune system. I took this dates me, I know, but I was taking undergraduate immunology course at the University of Texas at Austin. The professor, Bill Mandy, he was an antibody guy, but he had one lecture and he talked about T-cells because they'd just been discovered and basically about all they knew. I said, well, what about these T-cells, you know? And we went to see him in his office after class. He says, well, they just go around the body and, you know, go through your tissues and your blood and your lymphatics and do stuff, you know? And I said, well, how do they know what to do? How do they know, you know? He said, I don't know. He said, I don't even know if they really exist. I think they're just cells that pick up antibodies on their surface. And so I decided at that time I was going to figure out how T-cells worked. And that was a long time ago. Anyway, but still doing it. But it is an example of how basic science can really, I think that, you know, I still don't know what to exactly work, but I think that, you know, being able to be a basic scientist and see the work turn into a new whole kind of treatment for cancers, seeing my basic science turn into something that benefits people, I think is the dream of everybody, you know, and everybody who's in science. And I'm just lucky enough to had it work for me. And I think my second point, I think there's way too much emphasis placed now on all grants having to say what the relevance is to, you know, cancer. You know, because how do you know? You know, how do you know what's going to be relevant or not? You know, so who would ever thought CTLA-4? I know one thing, that nobody that was trying to discover how to use the immune system to treat cancer would ever have found CTLA-4, because there'd be no reason to look for it. You know, and so I think that, you know, people should just, you know, pick your problem, work on it, do the best work you can, and then try to figure out, you know, how we can use it to treat diseases. Anyway, with that, the CRI has been a big supporter the whole time, really. I've gotten support, and a lot of my fellows have gotten support. But I just want to use that as an intro to say, you know, of course, this work it really came from a large lineup of former students, both in Berkeley and Memorial Sloan Kettering, and then today at MD Anderson over the years. And it's been, you know, just an evolving experience. And the only thing I did with my hands was the first work on T-cell receptor. And after that, it's been somebody else's work, and you know, as a team. But that's also what it takes to get things done. But the inspiration from Lloyd and people like him that really taught us how to, you know, get into the clinic and what the goals should be. And I think we all experienced Lloyd's wisdom over the years. And you know, anyway, I'm happy to be here today.
Well, we're happy to have you, Jim.
And
I think we should have other people make some comments about what you think of Jim's discoveries and what has happened. So, Crystal?
Okay, well, again, I feel humbled to be on this stage because Jim is a true pioneer, a true giant, and I mean, he has just been such a role model for all of us. In many ways, what Jim's story is the best that science has to offer, and I want the people who are, the average citizen to understand why we need to invest in science. Having smart people like Jim be free to pursue a hunch that he thinks might turn into something someday or not. He simply thinks it's interesting. When you feed people like this, who have the talents, and you give them the opportunity, this is the kind of thing that can happen. You make fundamental discoveries, and it takes a long time, and you got to keep at it, but at the end of the day, you can cure people who are otherwise incurable. And this is why it's just so important that we believe in the power of science and innovation and technology, and we tell that story to the powers that be, who make a lot of these decisions. It's nonprofits, but it's also the federal government. And, everybody needs to understand how powerful this can really be. This is a perfect example.
Yeah, Crystal, I couldn't agree more. I think that the average person out there doesn't understand how science works. And I always say, as a head of a not-for-profit, and you're always raising money, and people want to say, well, how much money do you need? And you can't quantify that. I can't ask somebody and say, I need this much dollars, and it's going to cure cancer. Because science doesn't work like that. It's not linear. I think you have to just support really good people and give them the freedom to explore things that they want to explore that may go nowhere. And I think you can learn as much from failure as you do from success, and that's what science is about. And we need to have sustained funding in this country for science, because I think you can see where science can lead. And it's not overnight. I think always that's another thing. Return on investments, people want right now, come in and get out and get everything out of it. You can't quantify time either. It just takes good people asking good questions and doing outstanding science. Nina.
Well, I'd also like to congratulate Jim. I want to just emphasize that the impact of this award today cannot be understated. This is a dramatic, I think, recognition of Jim's work for the past several years, and highlights how someone who's an incredible scientist has devoted his life to understanding a mechanism which has had enormous impact for thousands and will be tens of thousands, if not hundreds of thousands of patients in the future. His work really underlies the fact that for the first time there have been developments in cancer immunotherapy that have really cured people, dare to say that, allow people to live many, many years, and we will see the impact of this discovery for decades to come. He's laid the foundation really for the whole field in terms of developing many, many new agents, and as a result has emboldened not only people in our field, but in industry and other sectors to jump into this field, put a lot of money, investigation in science. And it's a thrill, and I think for those immunologists who've been working on this for decades, and I hope some of your magic rubs off on and have had to face the field when no one thought really, except for a tiny percentage, that the immune system would have such an amazing impact on cancer. It is just so refreshing and rewarding to see Jim honored in this way, and for those of us who are continuing to work in this field, to see where it will go. It's a real thrill.
Well, I also, for those that don't understand, I think prior to Jim's work, so much of the effort was about how do you activate your immune system to recognize and destroy cancer. And I think there was a sea change, and it was a paradigm shifting when Jim found, really discovered it wasn't all about activation. You needed to deal with how do you get over this immune suppression, these breaks that are naturally there in your immune system. And that's a fundamental change that has really, I think, affected across everybody that's doing immunology research, that you certainly need activation, but you have to deal with immunosuppression. And it's not an insignificant thing. And this is, where I think the field has gone, and it's just, having been at CRI for 31 years, I never thought I would see this day that the Nobel Prize would be about cancer immunotherapy, that we would be really curing some patients with immunotherapy, and that it's working. So again, I just want to talk. I know I have Phil Greenberg in the audience, a longtime friend of Jim and colleague, and I'd love for you, Phil, to make some comments on Jim's unbelievable prize here.
Yeah, well, I do know Jim from when he was a much smaller guy. I still remember, we did our postdocs together in San Diego, and I still remember him driving around on a motorcycle collecting blood specimens to take back to the lab to try to figure out what an HLA molecule looked like. So it goes back a long ways, and it's been a great joy. And I think, people say that, luck is more likely to occur to the prepared mind. Jim was actually a little wild in that, I thought. Not so prepared, but always inquisitive and always trying to find something new. I think the work he did identifying the TCR had enormous impact. And he did that really virtually solo in a small lab in Texas, actually. And that was the beginning of a career where he started looking at T-cell activation and understanding how T-cells recognize something and how the response gets sustained, because it became very clear that just getting a TCR signal was not enough, and that led him to his original studies, studying co-stimulation with targeting CD28, and then eventually to looking for similar kinds of molecules, what else belonged in the family, and that's really how CTLA-4 came about. It looked a lot like a co-stimulatory molecule, and it turned out to be just the opposite, but so closely related that it's kind of an on and off switch. And I think it was his ability to not just do the basic science, but to recognize the potential impact and what these things mean, which is one of the things that makes him so unique. Because it would have been easy to just move on from that, but he actually saw that as a chance to understand responses that fail, and that was really the anti-cancer response, and he really pushed that. And I think, as Jill alluded to, this was not something that was readily accepted by the field. Certainly not. I mean, the science piece was, but the idea that this could ever be a therapeutic was actually pretty much discouraged for quite a while. And Jim had extraordinary perseverance. He first tried to collaborate with one company that was going to try to make it and bring it to the clinic, and they failed as a company, and Jim extricated his relationship with them and found another company that would. And I think, had he not been so persistent, we probably never would see CTLA-4 in the clinic, because it certainly had some toxicities early on that were difficult to understand and problematic. But again, I think we all understood that you can figure out what's right and what's wrong about something. And Jim is a testament to having done that. So it's been a great pleasure being a friend of Jim's for all these years and just seeing his star rise and rise. So congratulations, Jim.
Thank you. But you have to burn that picture of me in my bathrobe.
So after all of these accolades, I think we should talk a little bit about the conference, which was the original point of this press briefing. So I'll turn to our experts on this panel. Obviously, we know that the field has changed, opened up by Jim. We have a number of FDA approvals for a variety of checkpoint blockades, as well as for CAR T-cell therapies. So I think, where do we go from here? And Jim's already alluded to the need for combinations, but is there rational combinations that need to be done, and how do we get to them? So I'll leave that. Crystal, you want to go first?
There have to be rational combinations. This is the essence of what we do. And the point that Jim made, that the good news is that, the world is bought in. The biotech and big pharma, they understand the power of the immune system now. The bad news is now we've got hundreds of combinations, and frankly, we need to be more careful about, what is the bar for getting this, combination into the clinic. And so I don't think that there's anything new here. We just have to do good science, and we have to do the clinical trials that are based on the most sound preclinical model. And if we do that, and if we study the patient, then we will make progress. If we just throw darts at the wall and see what sticks, it's not going to get us anywhere. So this is, I think, this conference is giving us that kind of information about where the early signals are, why some combinations aren't working, and yeah, we have to believe that we'll be able to sift the wheat from the chaff.
Anything to add, Nina?
I would agree, and I think it's very exciting because we're going to start seeing combinations in the clinic, and a lot of iterative testing and design will be necessary to understand how these work. And I think what's very encouraging also is just the breadth of new technologies we now have to monitor responses at the single cell level in patients from patient tissue, and that I think will help us really understand how these different combinations work.
I also want to say a few words about the engineered T cells.
Yes, for sure.
Because we had a great session on that this morning, and I think it's really, this field is still somewhat in its infancy. We have 2 FDA approvals of CD19 CAR for 3 diseases, and more are expected. And what we saw today is that the power of genetic engineering to be able to engineer the cells to supply exactly the right tuning of a signal, to be able to interrupt inhibitory signals, to be able to give agonist signals. It just, the power of the genetic engineering, we are just scratching the surface. And so I do believe that we will see genetically engineered and adoptive T-cell therapies have activity in the hard-to-treat tumors like solid tumors. But again, we're going to have to be persistent. But we have lots and lots of tools in our tool chest that can be utilized.
So moving on to, I think we all know that it's not going to be one size fits all when it comes to immunotherapy, that it really comes down to personalizing the immunotherapy, giving the right immunotherapeutic to the right patient, and that's going to require biomarkers. So what do we feel is the most exciting advance there in finding biomarkers, which sometimes seems like a needle in a haystack to me, but...
So in terms of biomarkers, I think we already have a few, such as the expression of PD-L1 on tissues as adjunct biomarkers for intervention with antibodies, but clearly we need a lot more. And I think in terms of personalized therapies, understanding an individual's tumor microenvironment is going to be key, meaning what is the landscape of someone's tumors? What inhibitory molecules do they express? What activating molecules? What is the constitution of the tumor from the frequency, the nature of immune cells, and the tumor cell itself? So I think getting a better understanding of the tumor landscape, developing tumor atlases, combined with investigation of a patient's blood, will lead us to development of biomarkers in conjunction with clinical trials.
Jim, you want to comment?
Yeah, I mean, I agree essentially with all of that. I mean, even with checkpoint blockade, what James Sharma has found in our immunotherapy platform at MD Anderson is just an example of how when you give a drug, everything changes, and you need to know what that change is. For example, prostate cancer is very cold. There's very few T cells in it. It looks like pancreatic cancer, but if you treat it with IPI, it looks like melanoma. But the problem is there's PD-L1 all over the place because you've also induced PD-L1. And so that led to us to go to Mr. Squibb and propose a combination trial, which will be reported on sometime this fall, rather. And looks really good. But one of the things that we noticed was that in inducing PD-L1, it also resulted in expression of VISTA on a subset of macrophages, and not the same macrophages that made PD-L1. So that's a little complexity. But to get back to personalized treatments, that doesn't happen in melanoma. You don't see VISTA in melanoma. You see it in prostate cancer. Mm-hmm. So we're going to have to go through and really, again, not just look at baseline with the goal of trying to identify some predictive markers. What we need to do is look at baseline, look how it changes when you treat with one thing. That'll tell you what the next thing should be. But that may be different for different kinds of cancer in different individuals. So, it will have to be personalized, even with these things that are pretty broadly useful. And that's going to take, it's beginning to make sense. But, finding the ipi-nivo right now is the most powerful combination for sure. But, melanoma is 60%. Patients are alive 3 years after treatment. And so, the things to get it up to 100, and how do you do that? Well, that's the next question, Which one of these things is going to block which chemotherapy, how do you do radiation with it? But back in prostate cancer, maybe we need to add VISTA antibodies to PD-1 and IPI.
One thing with, having Nina here, talking about combinations and cold tumors and trying to make them hot. I mean, vaccines, which I know, Nina, you've been involved with the vaccine development for a long, long time, and I think there's a renaissance in people's belief in vaccines. I think they were ready to throw the baby out with the bathwater a number of years ago, but seeing that now that we understand immunosuppression a little bit better, what role do you think vaccines are going to play?
I think the most exciting development in cancer vaccines is the discovery of what we would call neoantigens, which arise from a patient's specific mutations. And so the recognition, it seems as though we keep recognizing this over and over, but the idea that a patient's tumors, which will of course have its own breadth of mutations, can lead to a patient's very specific set of neoantigens, which can be targeted. And we're learning, as we learned today from some of the sessions, some of these are shared. So we can potentially have shared neoantigens that are targeted in vaccine. Some, of course, will be very specific and specific to the patient. But the exciting thing is the technology that's emerged to identify these antigens. We'll hear more in tomorrow's session about platforms to stimulate T-cell response to these antigens, and then the idea of combining these vaccines with other modalities. So I think for the first time we have rational approaches and rational targets that can be applied today.
And I guess another point to bring up, and I think will be touched on in the meeting, is resistance. Obviously, we have immunotherapies that are working in at least a minority of patients, and even when they do respond, sometimes people end up recurring, even though there are those that have durable responses. So the question of how do we deal with resistance and how do we understand the underlying mechanisms that lead to this resistance so that we can apply a therapy to it. I would like to know what people think of what's cutting edge, what's new in the area of study of resistance.
Well, we've heard a lot of discussion today about this entity of T-cell exhaustion. And, exhaustion means different things to different people. In many ways, checkpoint blockade is reinvigorating exhausted T-cells. And so that really illustrates the power of what a T-cell can do if you can overcome this problem of exhaustion. But we also know that checkpoint blockade is not complete in terms of its ability to do this. And so I think there's a lot of very exciting work happening, some of it in the engineered T-cell space, to understand the biology of T-cell exhaustion and to be able to come at it from completely different angles. Rather than the checkpoint receptors, there are also other intrinsic features that mediate exhaustion in T-cells, and we're still diving deep into understanding those T-cells that Jim was talking about from how many years ago, but there's still a lot more we have to learn about how they become less functional and how we can reinvigorate them, and there's some very exciting work in that area.
And if I could add to that, in addition to T-cell exhaustion, resistance at the level of the tumor cells, so learning to escape recognition from T-cells. We're understanding more about that, losing class 1 molecules or key signaling pathways that allow a tumor to evade recognition. We're also getting a better understanding of the tumor microenvironment from the non-tumor cells that are also part of the tumor, including the stromal cells. We heard yesterday about how stromal cells can contribute through making factors that really basically shut off the immune system or prevent access of the immune system to the tumor cells. We're learning about non-T-cell immune components such as the myeloid cells, the monocytes, macrophages, which can in turn be dysregulated to become suppressor cells that can turn off the immune system. So it's an exciting time, and I think more discovery of these resistance mechanisms, especially by following a patient's tumors individually. Crystal alluded to the fact that we need to understand at individual level what's happening to someone's tumor with each intervention, and that in turn will lead to, I think, a lot more better understanding of resistance mechanisms.
Yeah, so I do think obviously a lot of focus has been on T cells here, but I think there are other immune cells and other non-immune cells that are obviously in the tumor microenvironment, and I think we've heard a lot of sessions at this meeting about that beyond the myeloid cells, but I think you're talking about NK cells and neutrophils, and so I think there's an awful lot of biology that still needs to happen, and very much so at the tumor site. And so I think this is another take-home message I would say to people listening that are not in science, the need for patients to understand the need for multiple biopsies. This is something that patients always, it's painful, who wants to have biopsies, but this is where we can learn so much. You need to understand what's happening before, during, and after treatment. And it's a very Immunology is very complex. There's a lot going on at that tumor microenvironment, and so you really need this precious patient samples to be able to do this and bring it back to the lab and understand mechanism. So, that's just a take-home message for everyone to understand the need for that and why you're asked to do that if you're in a clinical trial. Any other thoughts on topics of where we're going? What's the biggest areas to watch going forward, do you think? I know this is like crystal ballish. I get these questions all the time. I don't know what to say. Jim, future, where to watch?
I think that what we're going to see in the next 5 years or so is we're going to approach 100% in some cancers, patients responding, I think, by doing the right combinations. Not many, but at least a few. And I think that what we're going to see is, now that we've got 4 pillars of cancer therapy, immunology is the only one that can work nicely with the other ones. And the other ones are pretty much siloed, always have been, and always will. But now that we understand, a lot about immunotherapy and what's needed, the people with the other cancer therapy can quit worrying about killing every last tumor cell, just kill enough to let the immune system, take it out and start being really, really.
I think that's a great note to end on. We know immunotherapy can work with a lot of the other cancers, and I think, expect to see immunotherapy be around for a long time, and they're going to become standard of care for more and more cancers. So thank you all for being here, and for all those listening, and we will keep reporting on cutting-edge science that comes out of this meeting and beyond. And I think we're at a great place. And again, congratulations to our newest Nobel laureate, Jim.